Okay. Today I'm going to talk a little bit about our vaccine platform. First off, we're staying with the same theme. We really believe that oral vaccines are the way to go. I think, Rocky, you did a really good job of describing a lot of the ways that oral vaccination is really important, or it can be, game-changing. For us, one of the things that I also want to point out is that if you're trying to go into an existing market that's already very well saturated, such as influenza, you can make all kinds of great changes and improvements of the vaccine, but at the end of the day, if you're still stuck with a needle, then you still have the same sort of mode of delivery. And the only way to actually improve the uptake, considering that only 50% or 40% of the people actually take the flu vaccine, is to actually change the way it's distributed or administered. And I think from that standpoint, oral is actually the way to make this a different sort of mode of delivery. Vaxart, we're a very small biotech company, but we've done extensive market research, and I can tell you from our definitive market research, I can tell you mice don't buy vaccines, and so I'm hoping to sort of convince you that you should eventually buy our vaccine in the future. And so what I'm going to talk to you today about is human clinical trial work that we've done, and with a little bit of mouse and ferret work kind of sprinkled in. So a little bit about our platform. We are using an adenovirus. This is a very vanilla sort of approach, and of course adenovirus, it makes it very easy to change from one gene to another, sort of like I changed my 8-track tape in my car. So now you can go from an influenza vaccine expressing HA to an HPV vaccine expressing L1 very quickly. One of the things, when you deliver adenovirus to the intestinal environment, it tends to get ignored. It's not very immunogenic in this environment. It sort of acts like food. And so what we've added essentially is a double-stranded RNA hairpin. And what that does is essentially tells the immune system to respond. And this is a molecular adjuvant. And the advantage of taking this approach for an adjuvant is by putting it on the same backbone is that when we go into target cells, the only cells that actually see the adjuvant are the ones that are actually chock full of the antigen. And because we're using a non-replicating system, the only thing that's really being made, or the two products that are being made, is a protein antigen of choice and your double-stranded RNA adjuvant. And we think this is a reason why we're going to get better selectivity against the gene of interest, and we also are getting a much benign, so essentially there's no inflammation in the intestinal environment. A little bit more about the approach we're taking. Adenovirus is not stable in the stomach, and so we've actually used some pharmaceutical techniques for using enteric coated tablets. And I have some of those here as well. I'm going to show it to you in a few minutes. The other thing, is that the way we think this works is that, the enteric coating really protects it in the stomach. It's intact at low pH. At high pH, it opens up. We think that the way it works is actually it infects an epithelial cell. That epithelial cell then makes, again, only two products, our double-stranded RNA, which is the adjuvant, and fills it up for the antigen of choice. For influenza vaccine, that's HA. We think a dendritic cell comes along and swallows that infected cell and sort of makes a cross-priming approach. And again, because of where the adjuvant is recognized on that endosome, we think that means a much better selectivity. I'm going to tell you a little bit more about tableting. This is not, I guess this is unusual vaccine candidates, but certainly this is not unusual to make tablets in a pharmaceutical method. In fact, there's billions of them made every day. But there are some advantages from the standpoint of a vaccine. First off, we don't have to worry about sterile fill finish. Our product is actually not sterile at the end. It's a sanitary product. It has to have the low bioburden, and that makes it a huge advantage. We don't have to go through the same sort of purification, and it's a lot cheaper to do. And tableting, we can do millions of doses a day. Our biggest problem right now is finding a tablet machine that can actually make small-scale batches, because usually when you flick these on, you're basically in 10 minutes you've made 1,000 tablets, and for small phase 1 trials, that's really difficult to actually to have enough material to use. So we've made a lot of sort of improvements in the formulation, so it's room temperature stable, and we've actually have a very nice formulation, and now we're getting to the point where we have tablets. Ironically, we put them in a syringe vial. I haven't figured out how to get them out with a needle, but I can tell you that these things are pretty small, very easy, and room temperature stable. And certainly when we put them in this sort of vial, they're protected from moisture, so they'll last forever if I can get them out. So let me just explain what we've done. Again, we have done phase 1 studies on seasonal influenza using H1N1 monovalent approach. We have just completed a coated tablet study, and I have, unfortunately, not all the data, so I'm not going to be able to share a lot of that with you. But we've also done some work with, which I think from a scientific standpoint are actually very interesting, because we've used these radio-controlled capsules. And the nice thing about these things is essentially they're giant capsules about this big, and you fill them full of liquid vaccine and a radioactive tracer. And people actually have to get tested to be able to swallow this, but once they swallow it, you can kind of trace it, and you can actually ask questions about what part of the intestine is better in terms of eliciting an immune response. This technology has been well used in the drug development we're like the first to use it for a biologic, so it took us a little while to get FDA to be convinced, at least the biologicals division, to let us go through with this. And I think the nice thing, again, from our standpoint is, one, you can actually ask some really great questions about location, and the other thing is because you don't have to worry about this tableting problem of downscaling, you can actually make a lot of small-scale batches of your vaccine and really ask some questions before you go forward and start making your phase 1 material with the coated tablets. So anyhow, this is not a commercial product. They're $1,800 a pop, but it's very interesting kind of questions you can ask. The first slide is a very, what I would call non-scientific sort of like summary of the data in terms of the different sites of location and 2 different dose levels. This is again with the radio-controlled capsule technology. If you can look over here to the left-hand side, you can see that when you use 10 to the 10th at the first site, this is actually in the lower part of the small intestine, and compare it to a higher dose at 1E11, what you can see is in terms of total antibody responses by ASC or ELISA, as well as my neutralization titers by microneuter HAI, you can see there seems to be a dose response, and you can get up to 100% responders essentially, at this site at 1E11. If you deliver the vaccine actually to a site that's further up the intestine, you actually don't get the same sort of response at the same dose level. It seems to be less. So from this data, we were actually able to make improvements to the tablets to sort of target the right areas and move this product forward. So the next few bits of data I'm going to show you is mostly about the preferred site of location. So one of the assays we use for total antibody response is an ASC assay, and the best thing about this assay, there's 0 background. Almost everyone has been exposed to influenza before, and certainly background is an issue. But if obviously when you're looking at the antibody secreting cells that recognize HA, day 0 there's nothing there, whereas at day 7 you can see the responses of the vaccine. So this is a very nice assay to really teach you about your vaccine. I think what you can appreciate if you look at 1E10 versus 1E11 is that increasing dose leads to much more reproducible high level of total antibody, or B cells that recognize HA. Seasonal vaccines from our hands get ASC counts between 200 to 400, so we're doing pretty well. HAI is actually one of the surrogates protection for influenza. We certainly are advancing our product on an HAI sort of approach. This is a very individual subject sort of plot. We're looking at the day 0 versus day 28. What you can appreciate is every single subject responded in terms of increasing HAI titers. 67% were seroconverted, and at the end of this study, we had 75% seroprotection. The geometric fold rise is decent. And so again, from an HAI standpoint, the vaccine seems to be working pretty well. We also looked at neutralizing titers by micronute. Again, what you can appreciate is that 10 to the 11th versus 10 to the 10th gives you a much more reproducible response. And what I would say is that now we're at 90% of the subjects, or 92% of the subjects essentially in this study, having a neutralizing antibody response. Again, this is using the radio-controlled technology. If you look at this in a comparison, I'm using a paper that was published by the U.S. military, so it's an independent study, and looking at they buy a lot of FluMist and Afluria, or at least in this season they did, and what you can see is the micronute 4-fold rise rate for the injected vaccine versus FluMist, and then geometric titers. What you can see is we sit sort of in the middle. I can't really say statistically whether we're better than FluMist or worse than Afluria, but certainly we sit in the middle. So this was great for us. We thought this was a great step forward, and we actually, again, started this tablet study, and I'm going to show you the one piece of data from that tablet study that I have, and essentially what we're looking at the micronute response rate. Again, we're looking at with the 4-fold rises and the geometric mean titer at the end of the day, And in comparison to this Floria study, we're at 92% of the subjects had a fourfold rise. The geometric mean titer at the end is 247. And again, this is using the same sort of methodology with this independent study. So from our standpoint, it looks like we're doing pretty well now in terms of a neutralizing antibody response to influenza, at least in this H1N1. So now I'm going to talk to you a little bit more about just what the platform is. I'm going to talk more about the mucosal responses. Certainly, we're using an advector approach. We're delivering it to mucosal surface. We know we get good T cell responses. We know the antibody diversity looks pretty good. But now I'm going to tell you a little bit about the mucosal responses that we're seeing. So again, I guess I had one more tablet study, but anyhow, here's what it shows: the placebo versus 10 to the 9th, 10 to the 10th, and we're looking at the antibody secreting cells of IgA class, and this is compared to the injectable vaccine. This is your standard flu vaccine off the shelf, and we're looking at the number of antibody secreting cells. What you can see is there's obviously no background, which is why we like the ASC assay, and you can see that there's increase. You can find the antibody secreting cells at increasing numbers at increasing dose. And again, compared to the injectable vaccine, which aren't known to make a great IgA response, it looks quite nice. If you look at the 10 to the 11th dose, which is in the RCC study, what you can see is 100% of the subjects have an IgA ASC response. And again, the numbers and quantities are much greater than it was at a lower dose. One question we have, of course, is, just because we're making an antibody cell, and again, we're looking in the peripheral blood 7 days after, we don't know if these cells actually home to the bone marrow or they home to the mucosa. So the next question was to ask, do these cells, well, first off, we couldn't actually take biopsies of our subjects in humans and ask, if there's antibody secreting cells in the various mucosal compartments. But what we could do is ask if there was actually a mucosal marker on these B cells. And so we did a little bit of flow work. What we found is that after immunization, that a population showed up that is beta-7 high and alpha-4 high. Again, alpha-4 beta-7 is a mucosal homing integrin, or at least it seems to behave that way. And this population showed up, and up to 27% of the B cells essentially had a beta-7 high signal. If you gate on that beta-7 high population that exists now only on day 7, what you can see is it's 80% surface IgA positive, and it's larger and it's got more higher fortified and side scatter. So that this sort of data suggests that it's a pre-plasma B cells that would home to the mucosa. And if you look at the population that's beta-7 low, which existed before immunization, very few of those cells are surface IgA positive, and they're smaller. These are probably memory cells. I'm going to take a look at this in a slightly different manner. We're going to look at first CD27 expression, which is a marker of activation. Day 0, you don't really see much of a population. It shows up quite nicely in the peripheral blood, and this is again in humans. At day 7, if you gate on that CD27, the activated B cell population, what you can see is that, again, 85% of it is beta-7 positive, and very little of it is, negative. What we also found is not only is there a beta-7 population that shows up, but it also found a population that's beta-1 positive. These cells are also CCR10 positive. We believe that these ones may be actually homing to the nasal mucosa rather than alpha-4 beta-7, which would home to the intestinal mucosa or the cervical mucosa. So the question, is, can we actually detect a fecal or a nasal nasal sort of IgA response? And one of the problems you have with humans is they've all been exposed to influenza, so your background is going to be high. But we went and tried to do this anyways. So in the subjects that actually came in with the RCC, we were able to actually take fecal samples. I don't know how fresh it was, but what you can appreciate is that, it's actually hard to do in humans. Their patients don't really like to comply and bring your poop on usual schedules. So. But anyhow, what you can see is you can appreciate is that several subjects definitely have a detectable response in terms of an anti-HA response in the fecal samples, but not all. And certainly, the background I can tell you is actually really high in a lot of these samples. If you look at the nasal wash, this is there's a little less crud in this, and it may be easier to detect. But what you can appreciate at both either site 2 or site 1, about 5 of the subjects out of the 12, you can detect a shift in the anti-HA response. Again, the mucosa data from the flow looks like we're making a very large population, whereas in the mucosa samples where the background is high, you can detect some changes, but certainly doesn't look like it's that large comparatively. I think we're just underestimating, truthfully, the response. We also looked at whether T cells actually have a alpha-4 beta-7 marker on them, and unlike the B cell population, where a huge proportion of the cells are alpha-4 beta-7 positive after immunization, in T cells that would probably be deadly. So what we did is we actually looked in the cells that are actually responding to a peptide library and making gamma interferon at day 7, and then asked the question if we were enriching for alpha-4 beta-7 compared to at day 0 samples, and the answer is yes. We can find a population that seems to be alpha-4 beta-7 enriched in the ones that are actually making a gamma interferon response to HA peptides. So where are we going with this? Obviously, we like to think this is a good platform, and so we are actually starting projects where we're taking advantage of the T cell responses that are homing to the mucosa, which is our therapeutic vaccine and herpes, and we're also starting a norovirus project where we're taking advantage of the idea of making a strong B cell response. The last thing I'd like to leave you with is actually something that's really sort of, I would say, messed up the sort of vector-based approach to immunology. And that is the answer is the antivector response. These vectors work really well when you basically do this in mice, but they don't sort of work as well in humans, and they can't really reuse them. And this can be illustrated in this plot. This is in mice and ferrets on the right-hand side. And if you inject an ad vector that expresses HA, you make a very, very strong response against HA, and these would be 100% protective in a challenge model. What you also do is you also elicit a very strong anti-vector response, and that is in red. And again, it happens both in mice and in ferrets. If you take our approach with this molecular adjuvant and you have sort of like a very strong response against your antigen, again, you can see that really clearly at day 3. But what you don't seem to get in ferrets and mice and ferrets is you don't get that red response. You don't get the anti-vector response. And so when you actually try to use this vector again, it makes it very easy compared to when you use IM injection. That could be illustrated in the next slide. Again, here we're trying to use either a vector that expresses Venezuelan equine encephalitis first, and then following it up with a vector that expresses HA from influenza. If you have not used this vector once before, it does pretty well. If you've used it once for some other indication, this is in the IM situation, it doesn't work as well. You don't make as strong an immune response against HA. In contrast, if you do that same experiment orally, again, you don't seem to see the same loss in activity. It seems like the animals have never seen the virus before. Well, mice are nice, ferrets are good, but what happens in the human situation is actually important to us. If you inject adenovirus over here on the right-hand side, and you have a immune response against Ad5, what you can see is that 100% of the subjects actually seroconvert, and you make a very, very strong anti-vector response. Throughout the use of all of our studies, what you can see is that there doesn't seem to be that strong an immune response against the vector. We see a little bit of signal here in placebo and in some of the groups, but we don't see the same sort of 100% seroconversion and the great increase in anti-titers to the vector. And again, from our standpoint, in reuse and making this a platform, this is really critical. And I think this is a very nice piece of data from the standpoint of it's in humans, and it really shows that there's a big difference between this and an injected ad situation. So last, I just want to leave you with, again, we are developing oral vaccines. We think this is really important. We think, for indications like flu, it can make a big difference in sort of changing the market and increasing the uptake. We have all the data that suggests this is still a true platform. And from the manufacturing standpoint, we think there's several advantages using oral delivery. Lastly, we've shown a little bit of our phase 1 safety or actually immunogenicity data. The safety looks good. And I will be, once the data is actually complete for the tablet study, I will be presenting that, and that will probably happen at the World Vaccine Congress. We're almost done with the data. And lastly, I'd like to actually acknowledge the people that did most of the work, and especially Lisa and Kim, who did almost all the mucosal work in humans. It was a dirty, thankless job, and she took it on wholeheartedly. Anyhow, thank you for your attention.
So great talk. I was just wondering, could it be that you're inducing tolerance to the adenovirus vector, given that you're not seeing an increase in response? And then what would that mean in terms of if someone gets exposed to a pathogenic wild-type adenovirus? Would that actually make them more susceptible?
Yeah, I doubt its tolerance, but I can't exclude that. From our standpoint, it would probably be fine because adenovirus 5 isn't really a very pathogenic organism. So our sense is I doubt its tolerance. I think it's just ignorance. Truthfully, I don't think the immune system actually notices ad until the adjuvant actually is expressed, just because of the way the intestine sort of works in humans. And then when you turn on the adjuvant, the only thing it sees is the antigen HA. That's what I think, but I don't have any data to back that up.
Yeah, because it's a little bit unusual, because again, I still don't understand how the adjuvant, adjuvants increase the response to every protein.
Right. Well, think of it this way. Think of it as a kinetic argument. So you have your adenovirus and you have your target cell. Adenovirus gets in, it unwraps all the, the coat proteins. Those sort of like dissipate. It gets inside the, cell. The cell makes two things then from, at least from the vector. It makes the antigen of choice, HA, which it puts on the surface, and it fills this thing full up the double-stranded RNA, the danger signal. That's the point where the immune system sort of pays attention. And what does it have to grab onto? Well, HA is the obvious thing, so that's what it grabs onto. By the time, it basically, the immune system sort of wakes up, there's no coat proteins, or very little. That's my argument, and I have some data to back it up, but again, it's supposition, so.
Yeah, it makes sense.
It makes sense, but yeah, sorry, go ahead.
I'm just wondering, when it comes to mucosal vaccination, it is quite crucial that you choose the route of vaccination, as the pathogen is naturally entering the host. So in response to influenza, one would rather guess to take the intranasal route than the oral route for vaccination. And,
right. So we can find nasal responses, and it's not.
But you have given orally the
yeah, we can find antibody responses to HA in the nasal, but the correlate of protection and where everyone's going for approval on, if you want to have a very reasonable size phase 3, is HAI. That's an antibody IgG response in the serum.
But I'm talking about local responses as well.
Oh, I'm not local response. But flu probably doesn't need to have that response. But certainly for something like norovirus, we think that it'll be very potent in the intestine and may work better that way. But yeah, local is probably better.
Yeah, and the circulation of migrating cells would rather go into the target organ. That's
true.
That the mucosal
but the mucosal system is a little sloppy, at least in what we've noticed. And so, well, in humans at least, it's sloppy. In mice, it's very sloppy. So what we found, at least, you can find nasal responses. It's not as great probably as in the fecal, but you can find it. And certainly the FACS data would suggest that it's going to be more targeted toward the intestinal and the cervical mucus rather than the nasal. But we certainly find HA, so.
Did you look for the sublingual route as well? Because that is quite a potent route as well.
Yeah, we're not interested in sublingual because we don't have any IP on it, but just to be frank.
Immunologically, it would make sense.
And this looks so much nicer. Look at this. Look at this.
Still, the common mucosal immune system is not that common as one would guess.
Yeah. Okay, go ahead.
Okay, so if we have an orally delivered flu vaccine tomorrow,
yeah.
What would the cost look like compared to the current route of administration? And I ask that question because
it's important.
Yeah, it's important for flu. For flu, it is. So
flu vaccines, I think that the cost to make, and somebody from Sanofi will probably correct me, is about $1.50 for quadrivalent, and it includes probably about 50 cents of that is probably the fill finish of the syringes, right? The administration cost for you when you go to your doctor, I think, is about $30 to $70. So today, in a phase 1, we can't make this for less than $1,000 a dose. But in scale up, when you get to the scale, we can make it for about $1.50 for a quadrivalent. That's what we think. So when we get to the scale, we can do it cheaply. In phase 1, you can't. The waste in my tablet machine, I have to use 80% of my material sometimes. So it's just not very efficient at phase 1.
I think people would rather not have a needle, but the question is, are they going to pay?
Right. So I guess the thing is, if I can take the money that's given to the doctor, the $30 to $70, and put it to my profit, then I think I make it out way ahead, especially if I can make it for about the same cost. So, I mean, just to be, I mean, I'm here to sell vaccine, so.
Some vaccines, though, for flu, for example, I've had it in the pharmacy, so I walked into our pharmacy, and the pharmacy was
Right.
there's no additional charge. I don't know if they charge insurance for it.
I think that there's administration costs for that anyways from the pharmacy. And from my standpoint, I'm kind of busy and lazy, and so I don't like to wait. So I don't like the pharmacy approach usually. But it's better than going to my doctor, which I can never see. So, sorry, go ahead.
Yeah, actually, well, I'm from Sanofi, so you're pretty close with the numbers there. I think increasing coverage rates is a good point to mention, especially in children where intramuscular may not perform as we would like. So I think that's right.
Right.
But I wanted to ask a question about the kinetics of the response by this route.
Right.
And so, because, I mean, people understand that even with intramuscular immunization, you get kind of almost a waning of immunity even through the season.
Sure.
You know, especially at the tail end. And so I wondered whether for something like this, whether the kinetics were a little bit different, both at the front with kind of time to induced immunity, but also at the back end as well. And so how far does it go out? Does it go better than intramuscular and it doesn't taper off by the end of the season? So what does that look like?
In our experience in animals is that we have better durability of that antibody response over time. For some reason, the Advector sort of approach leads to sort of longer. I mean, that's our experience, but we haven't looked yet in humans. Our 6-month time point is coming up in another, well, for the tablet, not for a while, but for, this RCC study, not for a few more months. And so then we can look and we can ask the question. We know that flu vaccines wane pretty substantially over 6 months. It would be nice to see some durability. From my standpoint, I hope to at least equal the performance of the injected. That would be a good goal for us. So,
just as a quick follow-on, I'm sorry, but it would be of interest even under a scenario where you would look at prime boosting as well, because if you have a good response orally, and then you can come back even with a parenteral boost, even with a, dare I say it, a subunit, but
right.
Such as inactivated or whatever, actually, it could be even.
If you really want to, maybe not for flu, but if you, for some reason, you really want to focus a very strong antibody response, priming with this and then probably coming back with a protein boost would probably really give you a very strong antibody response in whatever your protein. And again, because the protein is folded already in the same conformation, you'll go after whatever the best antigen is. So from that standpoint, great. For flu, maybe you want a little more slop to give you cross protection, but again, it could work still.
Yeah, cross protection you can get to in various ways. You can tailor your antigen. You can also look at getting heterosubtypic protection by changing the modality, the delivery vehicle itself.
Right.
That's fair enough.
Yeah. Okay. One more question. Sorry, is somebody next?
Sorry.
After me? Okay.
Yeah. Oh, I was also not convinced by your nasal responses.
Yeah.
I think the intestinal responses are convincing. Seems to be convincing, but this is really not a proof that there is happening a local immune response in the nose,
right?
Or in the upper respiratory tract.
Yeah, and the big problem is that I've found is that everyone's got flu or has had flu, and there's already pre-existing immunity in the nose. And so what you're trying to do is you're trying to see if there's any vaccine improvements. The best thing I could do is basically I could go and start teasing out this sort of these beta 1 and start looking at the markers and look for those cells that are being appearing. But or I have to go with an antigen that where people haven't been exposed to it. Maybe the Ebola vaccine, I can look at nasal responses and see if we get a better response. Because, obviously everyone's had flu. The background is really high in the nasal washes. And in the ones you can actually see it, or the ones right here where you see it, that's probably because the background just happens to be lower in that subject.
The problem is that measuring IgA in the nasal secrete isn't telling you anything. Because it's an overflow of what is in the serum. It's not local. It is not per definition locally produced. So if you
I would disagree with that assessment, and I'll talk to you about it afterwards. But